Okay, so welcome everyone to this month's edition of the Optics Interest Group seminar. And so today we have our very own speaker, Lina Streich, who's currently a bridging postdoc in our lab. So Lina has studied biophysics at Frankfurt University for her master's thesis and has specialized in ultrafast spectroscopy during this time. And she was interested in studying vibrational energy transfers in organic compounds. She then became very interested in applying those skills to a more biological environment, and that's why she joined my lab in 2016 as actually the first PhD student of the lab. And she started to work on multi-photon microscopy with a particular interest in adaptive optics. And I also want to mention that since last Friday, Lina is also a PhD, freshly defended. So congratulations once again. And I'm very happy that today she will talk about her thesis work and about three-photon microscopy and adaptive optics. So please, Lina, the screen is yours. Go ahead.
Yeah, thanks, Robert, for the introduction. Okay. Yeah, and also thanks for the organizer of this joint optics seminar for giving me the opportunity to speak today. And as Robert mentioned, so I did my PhD in his group, and I will present you the work of the last 4 years where I developed the three-photon microscope in combination with adaptive optics to enable really high resolution imaging deep in the brain. And to understand the function of the brain, how it is connected and how it processes information has been of great interest to scientists and society over decades and centuries. And however, in most cases, we cannot get access to the human brain, and therefore the mouse as a mammalian model or organism has become a widely used model organism in neuroscience. And as optics people, you probably know that ideally we would have an imaging technique available which would allow us to record the entire mouse brain at synaptic resolution and at volume rates comparable to action potentials. However, we do not have one such technique available, which allows us to recover all of the required parameter space in terms of imaging speed, field of view, achievable imaging depth and resolution, but we rather have specialized microscopes. So the aim of my PhD was to push the frontier of microscopy with respect to achievable imaging depth, resolution, while imaging the in vivo mouse brain. And for imaging inside scattering tissue, multi-photon microscopy has become the gold standard imaging technique. And in multi-photon microscopy, a fluorescent molecule is excited with 2 or 3 photons of double or triple the wavelengths, so that only due to nonlinear excitation, only at the focus, the fluorescent excitation density is high enough to generate fluorescence. So multi-photon microscopy has inherent optical sectioning, which allows to detect both scattered as well as ballistic photons, significantly increasing the detection efficiency. But it also allows to shift the excitation more to the near-infrared spectral range, where scattering is generally reduced, so also penetration into the tissue enhanced. But also with the good 2-photon microscope, we can currently only investigate the upper layers of the cortex at high contrast to resolve fine structures such as the small protrusions of the glia or fine structures of the neurons such as the spines. Although 2-photon microscopy can be pushed towards a millimeter, however, with the trade-off of having reduced contrast due to the onset of out-of-locus fluorescence, which you can see in this image here, that large structures are still visible, but you really lose the contrast to resolve the fine detail. But with three-photon microscopy, which was first demonstrated in 2013 by the Chris Chu group, due to longer wavelength excitation and to inherent improvement in contrast, you can image much deeper into the brain. And here you can really appreciate, and in the 3-photon image compared to the 2-photon image at the same imaging depth, you have superior contrast to resolve really the fine details. So why is 3-photon microscopy advantageous for real deep tissue imaging? First of all, it also allows you to shift the excitation even more to the near-infrared, where scattering is reduced. But if you think about maximum penetration depth, you have to consider both scattering as well as absorption, which is mainly dominated by water, which results in 2 optimal spectral windows around 1300 nanometers and 1700 nanometers. So these 2 spectral windows could also be used for 2-photon excitation, more near-infrared fluorescent probes. However, the second big advantage of 3-photon excitation is the fundamental improvement in signal to background ratio due to later onset of out-of-focus fluorescence. Because if we go deeper into the tissue, we need to increase the power exponentially so that at a certain depth, the photon density at the surface is high enough so fluorescence also generated there, leading to loss of contrast at depth eventually. But due to higher order nonlinear excitation, this onset of autofocus fluorescence occurs much later for three-photon excitation, therefore yielding a fundamental improvement in signal-to-background ratio. And the second consideration we have in microscopy is image quality degradation due to optical aberrations. So in the ideal world, we would have the same refractive index between the optical materials and the sample, leading to a perfect diffraction limited focus. However, in the real world, we have refractive index mismatches, which actually leads to wavefront distortions and therefore also degradation of the point split function, especially in the axial dimension. However, as the accumulation of aberration is an entirely deterministic process, one can define an ideal modulated input wavefront, which allows to correct for all aberrations and therefore recover near diffraction limited resolution. So to come back to our aim, so to improve the imaging depth, we use three-photon microscopy, which can image deeper than two-photon microscopy due to longer wavelength excitation and fundamental improvement in signal-to-background ratio. We implement adaptive optics strategies to recover resolution at depth. And as we are imaging the in vivo mouse brain at high resolution, we also implemented intravital imaging strategies, which allow to correct for motion artifacts which result from vital functions such as the heartbeat. And we also had a special focus on the light exposure we put onto the brain, as we want to maintain physiological imaging conditions. So this is a schematic of the experimental setup and the microscope which I built during my first year. And so the absorption cross-section in three-photon excitation is orders of magnitude smaller compared to two-photon excitation. So this has to be compensated by optimizing a multitude of parameters, such as the laser repetition rate, the pulse duration, and the objective NA. And as you cannot arbitrarily ramp up the power because you have tissue heating, the workaround in three-photon micro excitation is that we use low repetition rate, high peak intensity, short pulse duration laser sources. So we're using a 400 kilohertz repetition rate laser source, which generates around 30 to 50 femtosecond pulses. And as we're working with these very short pulses, dispersion compensation becomes essential. So as the fluorescent signal is inversely proportional to the square of the pulse duration. And as a pulse compressor, we implemented a single prism pulse compressor, and some of you in Janelia might be familiar with this design, where you can either use the design with two roof mirrors or roof mirror and a corner cube. We actually chose the design, which is not shown here, with two roof mirrors, because there you can maintain P-polarization so that you can always go through the prism at Brewster angle, which increases the overall transmission through the compressor. So with the compressor in place, we can recover 50 femtosecond pulses at the sample plane, therefore increasing the signal 50-fold. And the resolution we currently achieve with the microscope is 1.8 microns in the axial dimension, which is in accordance with simulations I previously did in ZMAX, while this is with an overfilled objective NA. And I've also implemented adaptive optics into our microscope, and there we've actually worked with two different deformable mirrors. So both of them continuous membrane deformable mirrors, one from Boston, one from Alpao. And I also implemented different calibration strategies based on Schack-Hartmann or interferometer, and I will be talking about this later during the talk. So which new biological questions can we address with three-photon adaptive optics microscopy? So three-photon microscopy allows to image subcortical brain regions such as the hippocampus non-invasively, which previously required the removal of overlying brain tissue in combination with two-photon excitation. And correcting for aberration with adaptive optics allows to visualize smaller structures at large depths due to improved resolution and more efficient fluorescent excitation. And the same can also be essential if you're interested to investigate really heat or photosensitive processes, as aberration correction due to more efficient fluorescent excitation would also allow you to decrease the excitation power. So to establish our technique, and especially to evaluate the adaptive optics correction, we needed a resolution target deep inside the mouse brain. So spines, as these are small structures of submicron size, are an ideal such resolution target. So for first experiments, we used a Taiwan GFP mouse line, and to gain optical access to the brain, we used a cranial window preparation, which are done by Juan, a postdoc in our lab. And here I would like to show you some of the results with three-photon microscopy in the visual cortex and hippocampus acquired at 1300 nanometers excitation. And here in blue, you can see the third harmonic signal, which mainly visualizes vasculature. And in green, you see the neurons labeled with GFP. So with a good two-photon microscope, we would achieve high contrast CRISPR images approximately down to 600 micrometers in the cortex. But now with three-photon excitation, we can go much deeper into the brain, down to 1.2 millimeters. So yeah, here you can see the vasculature in the third harmonic signal. And here you can appreciate in green that we can detect the pyramidal 5 neurons in the cortex. We can clearly see the fibers of the corpus callosum. And we can also visualize the CA1 hippocampal neurons. And if we have a closer look at an absorbed at a cross section at layer 6 at around 700 micrometer depth, you can appreciate that even at this depth with three-photon excitation, we can clearly visualize all the fine details of the dendritic branches. We then go deeper into the brain, we can clearly see the fibers of the corpus callosum, which are the long-ranging axonal fiber bundles connecting the two brain hemispheres. And if we go deeper into the brain at over 1 millimeter depth, we can see the CA1 hippocampal neurons. And if we change the contrast, also here you can appreciate that even at this depth, we can still really visualize the fine dendritic branches. But remember, our goal was to achieve near-diffraction limited resolution at large depths, and hence also be able to resolve spines throughout an entire cortical column. And if you have a look at this zoomed image here of the dendrite, you can appreciate that it's rather difficult to tell whether there are spines on this dendrite or not. And that's why I've also worked on two complementary approaches, intravital imaging and adaptive optics, to improve visibility of fine structures deep inside the brain. And one of the challenges we are facing when imaging small structures in the in vivo mouse brain is tissue motion due to the heartbeat, which you can see on this video here, that due to the heartbeat, we have significant wobbling of the structure. So in principle, on a standard strategy to improve the signal to noise ratio is to perform frame averaging. However, if we now perform frame averaging with this high content variability, what we end up with is a complete blurred image. That is why we implemented the capability into our microscope to gate our image acquisition in real time to the cardiac cycle. And real-time data processing of the ECG signal is performed by an FPGA, which was programmed by Matteo and Ling from our group. So we can use the laser clock now to gate our microscope so that the dust tasks hold when our gate is high, so that really the scanning and the image acquisition stops, and that scanning only continues for the next high gate, so in between the heartbeats, where tissue motion is minimal. And here you can see another example of a neuron acquired at 700 micrometer depth, where without cardiac gating, you can also see that we have this really pronounced high frequency motion artifacts, which disappear when we enable cardiac gating, and we rather have some low order translational movements. You can also further quantify the frame-to-frame variability by standard deviation projection images. Here you can also clearly see that without gating, we have high content variability at the boundaries, which disappears when we enable cardiac gating. And we can also compare the frame-to-frame stability via cross-correlating individual frames to one another. And also here you can clearly appreciate that we have a much higher content stability with cardiac gating. So if we now want to perform frame averaging to improve the signal to noise ratio of small structures, you can appreciate that without gating, it's difficult to tell whether these blobs are actually spines or not. But here in the case where we enable cardiac gating, we can clearly see that we actually had spines on this dendrite at 700 micrometer depth. So heartbeat gating allows us to improve the signal to noise ratio of small structures by frame averaging, but it does not really allow us to improve the real resolution of the microscope, which can be done with adaptive optics. And in adaptive optics, we mainly differentiate between two main types of aberrations, mainly system aberrations, which result from the optical components themselves and due to the sample preparation, so the refractive index mismatch between the objective, the immersion media, and the sample itself. And correcting for system aberration yields ideal performance of a microscope. While sample aberrations can vary even across a field of view, and therefore correction for sample aberration, we need an active optical element which can modulate the incoming wavefront, such as a deformable mirror. But the main challenge in adaptive optics is to find the correct input wavefront. And there are two main approaches how to sense the wavefront, maybe direct and indirect wavefront sensing. So in direct wavefront sensing, and this has already been combined with two-photon microscopy to image or correct for aberrations in the in vivo mouse brain, which was mainly demonstrated by the group of Nagy and Kleinfeld. So in direct wavefront sensing, as the name suggests, the wavefront is measured directly, where the fluorescent light emitted from a point-like source, a so-called guide star, is detected by a Shark-Hartmann wavefront sensor, which consists out of a microlens area with the camera at the focus. And by measuring the shift in the intensity centroids, one can reconstruct the wavefront. So the advantages of direct wavefront sensing is that it is fast. It only requires a single measurement on the camera. And then if implemented in a D-scan pass, it can also yield rather large isoplanatic patches. But the disadvantage is that it requires detection of ballistic fluorescent photons and that you're actually measuring the aberrations from your emission light and not the excitation light. And this bottleneck, so that ballistic photons have to be detected, actually inherently limit this wavefront measurement with respect to depth, as you can see in this image here, where the guide star image becomes increasingly blurred and eventually at a certain depth, wavefront reconstruction becomes impossible. And so now in three-photon excitation, we actually excite at 1300 nanometers excitation. So if not the fluorescence or the emission of the guide star is also shifted towards this spectral range, which would require development of more novel fluorescent probes as well as detectors. Indirect strategies are the preferred options because there you really take advantage of the increased imaging depths in three-photon excitation and you correct for the aberrations of your excitation light. But of course, the disadvantage is, as it is an iterative approach, it's rather slow. And also aberrations are measured really locally, which potentially can yield smaller isoplanatic patches. So we implemented a indirect wavefront sensing approach because we really wanted to push aberration correction with respect to depth. And also in indirect wavefront sensing, there are different strategies to find the correct wavefront, mainly the pixelated piston-based approach, the zonal approach, and the modal approach. And which technique is most suited depends on several considerations. So before implementing a certain technique, one can ask, so how complex are the aberrations? Do I expect to have rather low order smooth aberration, or do I expect to have very complex, even some steps or some edges in my wavefront profile? Under which SNR conditions do I expect the wavefront correction to be performed, and how stable is the signal which I optimize, for example, the fluorescent signal intensity? And a nice comparison between the different techniques is actually summarized in this paper by Hu from the Bruce Group, which I can highly recommend. So in the pixelated piston-based approach, which comes more close to actually wavefront shaping, the pupil is divided into many small subzones. And In each zone, the phase is modulated so that the multiple scattered waves interfere constructively at the focus. While in zonal wavefront shaping, which can still be more considered towards adaptive optics, so where we still have a continuous wavefront which we want to correct for, the pupil is divided into intermediate sized zones. And in each zone, then the tip tilt, so the shift of each beamlet is corrected so that all of the beamlets intersect at the focal point, and subsequently the phase is adjusted so that all of the beamlets interfere constructively. While in modal-based correction, the entire pupil is modulated at once, and you use different modes with different mode amplitudes. So which technique works best under which scenario can be classified with respect to the signal-to-noise ratio and also how complex the aberrations are. So here on the x-axis, you have the amplitude of the aberrations, while on the y-axis, you have the complexity of the frequency component of the aberrations. And as one could imagine, so in pink represents modal approaches. These work best for more low order aberrations, while the pixelated or the zonal approaches have better performance when aberrations are more complex, as they can also represent steps, for example. But the situation changes if you consider low signal to noise conditions, where it shows that actually modal approaches are more robust under low signal to noise conditions, which is also intuitively, if you think about that, you modulate only a sub-region of your pupil, your expected change in signal, your modulated signal is smaller than if you actually modulate the entire pupil at once. And as we really wanted to push aberration correction in three-photon microscopy with respect to depth, we expected to have low signal to noise conditions and therefore chose a modal-based approach. And another consideration when choosing the optimization algorithm is actually the stability of the signal. Because we're imaging the in vivo mouse brain and we chose to use the intensity as a feedback signal, we can have intensity fluctuations due to motion artifacts, due to bleaching, but also especially due to intrinsic fluctuations, especially important for functional imaging. So we wanted to choose an approach which not only allows us to perform aberration correction on a stable structure, such as a GFP-labeled neuron somata, but also on structures which are, for example, labeled with GCAM. And in principle, the modes are dependent to one another, which means that they cannot be corrected independently, leading a large number of measurements which need to be performed. While this number assumes a full search, there are also more advanced algorithms such as hill climbing or stochastic algorithms, but still all of these algorithms require that the fluorescent signal stays constant over the measurement period. Well, if one now chooses an orthogonal basis set, this actually allows to decouple the optimization process so that the modes can be treated independently, so that in principle, the optimum amplitude per mode can be found within 3 measurements. And this can be essential if the wavefront correction has to be fast, if you are actually doing wavefront correction on intrinsically fluctuating structures. So in summary, we use an indirect modal-based correction scheme where we iteratively modulate the shape of the mirror and we take the fluorescent mean intensity as a feedback signal. And for wavefront modulation, we use Zernike polynomials, which are polynomials orthogonal over a unit cycle and present common aberrations such as coma, astigmatism, or spherical aberrations. And when working with continuous membrane deformable mirrors and modal-based representation, care has to be taken to prevent sample-dependent biases. So if we have a look at these 2 examples, so if you position your focal plane on the brightest structure of interest and you add aberrations, your metric value will drop so that the maximum value you obtain when aberrations are minimal. However, if you now position your focal plane outside or close to a region where there's a bright object, which can actually happen that if you add aberrations, you get a higher metric value because you push the point split function towards the brightest structure. And you want to prevent the sample-dependent biases, as this will corrupt your correction scheme. So while this is important to consider on which structure you want to perform aberration correction, and generally somata are the brightest structure within the region of interest, this is also especially important when you calibrate your deformable mirror. So I've worked with 2 different deformable mirrors from Boston and Alpao, and as I previously mentioned, we used an interferometer design for calibration, which was the design and the software provided by the Booth Lab. We calibrated the mirror with our own built Shark-Hartmann wavefront sensor and software, as well as the Alpao mirror was calibrated in-house. And in all cases, we always observed focus shift associated especially to spherical modes. So Z11, Z21, Z36. And you really want to analyze your modes, whether there are axial shifts to prevent sample-dependent biases, and if there are correct with this. And a correction of the axial shift can actually be performed by simply adding the opposite defocus mode, which works relatively well with small amplitude ranges, while for the larger amplitude ranges, due to the nonlinear character of deformable mirrors, this breaks down. So for larger amplitudes, I generated a lookup table which can automatically adjust the axial position of the microscope. And to give you an overview of the normal acquisition parameters we use for adaptive optics correction. So we generally acquire images at 20 to 30 Hertz, and generally I include 20 to 40 modes. And here, remember, as we treat the modes independently, I apply 9 different amplitude steps per mode so that the correction can be performed within 500 milliseconds. And this is how a general metric would look like, which evolves during optimization. So we have a clear increase in signal intensity in the first iteration, and we only get a minor improvement in the second and in the third iteration, which actually validates that our assumption that the modes can be corrected independently is valid.
I have a quick question.
Yeah.
When you're doing these corrections, are you doing a point correction, or are you collecting a vol? It sounds like you had frame rate on the previous slide, but I guess that's just the frame rate drawing to the DM, and you're doing point corrections here rather than collecting a little volume. Is that right?
No, actually, we are scanning. So normally with the 64 × 64 resolution, and then scanning over the entire somata, which mainly also in three-photon, even though, I mean, we significantly reduce the excitation power, but scanning still participates the power, so that we are not really like focusing onto one spot.
Understood. Thank you.
But you could also focus on one spot, and then you would be limited by the update rate of your deformable mirror. Yeah. So yeah, now I would like to show you some of the results we achieved with three-photon adaptive optics in layer 6. And here aberration correction was done on the somata, and generally the power is reduced during wavefront determination 50-fold, so that really only a dim signal of the somata is visible. And as you can appreciate, with full aberration correction, we can achieve a clear enhancement in signal intensity as well as resolution, where fine structures such as dendritic spines only become visible after aberration correction, which you can also see in this intensity plot here. But we can also have a look at an orthogonal view across a dendrite. And here you can also see before aberration correction, the point split function is significantly distorted, especially in the axial dimension, which can be recovered with full aberration correction, yielding a 6-fold increase in intensity and a 2-fold increase in axial resolution. And here's another example, also acquired at layer 6 at around 800 micrometers depth. And here you can really appreciate that with full aberration correction, we achieve spinal resolution throughout an entire cortical column, so that all of the fine details become visible. But we can also do aberration correction at over 1 millimeter depth in the hippocampus. And also here you can appreciate that we can significantly improve the axial resolution so that the distortions are corrected for, yielding to an increase in signal intensity so that the fine details become visible. And we can also quantify the improvement in resolution via Fourier transform analysis. And here you can also appreciate that both in the cortex as well as in the hippocampus, after aberration correction, we have a clear enhancement of the high spatial frequency component, which leads to an improvement in lateral resolution. And we can also quantify the improvement in axial resolution. And here you can see the axial full width of max plotted for different depths. And in blue, you see the axial full width of max before aberration correction, where you can clearly see that the point split function degrades with imaging depth. While in all cases with adaptive optics, when we can recover near diffraction limited resolution. So by combining three-photon microscopy and adaptive optics, we are now able to achieve near diffraction limited resolution down to beyond 1 millimeter depth. So with our microscope, we set out to also study a different cell type in the brain, glia. And glia neurons consist in the brain of a ratio about 1:1. And by now we understand fairly well of how a neuron processes information, but we know very little about the neuron-glia interconnection and how glia are involved in modulation of synaptic plasticity and transmission. In this project, we work together with the group of Amit Agarwal downtown in Heidelberg University, who is especially interested in astrocytes. And astrocytes exhibit a multitude of functions, and the star-like shaped cells branch out into the tissue and form thousands of contacts with synapses. However, unlike neurons, astrocytes are not electrically excitable, but they do exhibit calcium transients, which you can see in this video here. And the calcium transients of the astrocytes can vary in shape, but also in size, but also in space and in time. And for a long time, it has been thought that the calcium transients in astrocytes are actually too slow to play an active modulatory role, but that they are rather listening to the environment. However, it was recently found out that a subgroup of this calcium transient, which is associated to synaptic activity, can potentially be fast enough to play an active modulatory role. So these are on the order of 300 millihertz. So, but the functional significance of these so-called microdomains is still not fairly well understood. And if you actually have a look at the complex calcium patterns within a single astrocyte, but across an astrocytic network, this can almost change instantaneously. So while it is still not known or not clear whether astrocytes participate in real-time decoding of information, if this holds to be true, this would add a whole new layer of complexity of how the brain processes information. So most of the knowledge we have of astrocytes and calcium signaling in astrocytes is achieved by a two-photon microscopy in the in vivo mouse brain in the upper layers. And like neurons, there are different classes of astrocytes, mainly protoplasmic astrocytes, which reside in the gray matter, and fibrous astrocytes, which reside in the white matter. So protoplasmic astrocytes have already been investigated in vivo in the upper layers of the cortex, while fibrous astrocytes, due to their deep location in the brain and their location amongst the highly scattering corpus callosum fibers, have previously not investigated in vivo and non-invasively. So we set out with our microscope to study both deep protoplasmic astrocytes and fibrous astrocytes. And here you can see an example of a protoplasmic astrocyte acquired at layer 6 at around 800 micrometer depth. And here the astrocyte is labeled with GCAM. And here on the right, you can see the microdomains, which we detect automatically. So you can really, at this steps, can appreciate it. We can clearly see this blinking of the astrocyte. And automatic detection of the microdomain was performed via Cascades, which stands for Calcium Signaling Classification and Decoding, and which is an analysis pipeline developed in the group of Amit Agarwal. And data analysis was performed by Khalil, a master student in his lab. So in this example, we were able to extract 90 individual microdomains with a fairly good signal to noise ratio, as you can appreciate. But if we go even deeper into the brain, we were also able to record calcium transients from fibrous astrocytes, which to the best of our knowledge is the first in vivo recording of these cells non-invasively. And also with the fibrous astrocytes, we were able to detect 58 individual microdomains. And we made an exciting discovery that although the fibrous astrocytes reside in the white matter, so they do not tend to form regular synaptic contacts, they still exhibit microdomain-like calcium transients. But another characteristic of astrocytes, and actually a challenging one for microscopists, is that astrocytes are extremely light and heat sensitive, so they quickly change their physiology upon prolonged light exposure. And additionally, the calcium signals which are of high interest are the synaptic associated ones, so the microdomains which require high resolution imaging. So implementation of adaptive optics for recording astrocytes is really beneficial in two ways. First of all, it allows you to detect smaller structures. But secondly, correcting for aberrations and recovering for efficient signal excitation allows you also to reduce the excitation power and therefore maintain more physiological imaging conditions for the astrocytes. And here as a reminder, so here we're doing really aberration correction on functionally fluctuating structures. And aberration correction is performed on the somata, and the calcium transient of the somata is on average around 8 seconds long. So we are an order of magnitude faster to correct an independent mode where the signal has to constrain to be constant. And here on this video, you can see a layer 6 protoplasmic astrocyte at 800 micrometer depth. And here you can see the video before aberration correction and appreciate it's difficult to resolve transients. By now, aberration correction is enabled, and we can clearly see the transients traveling through the astrocyte. And this improvement in signal amplitude, you can also appreciate here on this calcium traces, where we have a clear enhancement of the mean amplitude of the signals, which is also plotted on this graph here. But even more importantly, we were able, with aberration correction, to improve the detection efficiency 6-fold. So before aberration correction, 9 events were detected, while after aberration correction, we detected 56 events. So with our adaptive optics three-photon microscope, we can now ask and study new questions in the field of astrobiology. We can now, for example, ask and study the differences of the calcium signaling between protoplasmic astrocytes and the previously understudied fibrous astrocytes, also in a layer-specific manner. The microscope can also be used to study structural changes, for example, spinal remodeling, also in a layer-specific manner, as we achieve synaptic resolution now through an entire cortical column, non-invasively and longitudinally. And I also believe that our microscope has the potential to study synaptic plasticity in the hippocampus in a non-invasive manner, because what currently limits us to go deep into the brain is actually the lack of excitation power from the laser. But the microscope can not only be applied to mouse neuroscience, but could be used across different model organisms and also in different fields such as developmental or cancer biology. And actually in another project together with the Prevedel Group, we studied mouse vasculature through the intact skin of the mouse, which is also highly scattering tissue. And with this, I'm at the end of my talk, and I would thank all the people involved in this work, and I'm happy to take questions.
Yeah, thank you very much, Lina. Very well presented, I hope. But I'm sure there's also more questions. So I wanted to remind everyone to sort of, for more interaction, to please raise your hand. You find the button on the bottom under reactions, and then to unmute yourself and post the question directly. Yeah, Boaz, please.
Have you measured, like, what is the size that you're, like, what is the field of view that you're correcting for?
Yeah.
so when you correct for, it was pretty impressive that you corrected for a soma in the middle, and spines at the edge of the field were corrected, which is amazing. Did you systematically try and investigate what is the SNR increase of a volume with and without AO?
I didn't systematically investigate this. So what I can tell, I mean, as I did several tries. Of course, the deeper you go, so in the hippocampus, certainly the correction is valid over 30 to 40 microns, I would say. And it starts to, in most situations, kind of then even at the borders, if you acquire a larger field of view, let's say 400, you even have decreased resolution. So because you're not correcting anymore for the correct aberrations. So that tendency, I definitely see that the isoplanatic patch decreases with respect to imaging depth. And in the hippocampus, I have to say there was a large variability, and I assume that kind of might also be dependent on, the preparation, right? If you have some curvature, if you have a larger vasculature close by, so sometimes the isoplanatic patch was 150 microns and sometimes also smaller. So that really depends, I think, on the sample preparation. So, but you definitely have that tendency that the isoplanatic patch decreases with imaging depth.
Thanks.
Caspar, please go ahead.
Yeah, I wanted to hear more about the ECG gating. So, naively, I would have thought to measure the ECG, collect the data, and then try and use that to align afterwards. But I don't know all of the considerations. If you don't scan during certain portions of the ECG, maybe you can apply more power, right, only during the phase that you care. Maybe you can tell us a little bit more about the decisions behind that and what the benefits are.
I mean, in principle, you could also do the post-processing. I mean, it's not in all the cases the same deformation. And then it's also a question, right, which signal to noise condition do you have for your really post-processing alignment? So there you really have the advantage that you align in the real image, so that even if you have really the very dim structures, such as some of the spines are not visible, you have a higher chance or better chance to overlap them correctly than if you do it under very low SNR images in post-processing.
Got it. Thanks.
Okay, and then we had another question from the chat. So Lina, can you explain a bit more how you correct for calcium transients when optimizing the wavefront?
So we are not correcting for calcium transients, but that consideration that we treat the modes independently. So in principle, you only need to perform 9 measurements or 5 measurements. So this can be acquired within 200 milliseconds, which is an order of magnitude slower than the calcium transient on the somata. So basically, just try to be faster.
I hope that answered Tim's question. Daniel, please.
You mentioned that you use the ALP-AO and the Boston mirror. What were the advantages, disadvantages of those two?
So the ALP-AO mirror they have roughly the same number of actuators. So the ALP-AO has 97 and the Boston 140. The ALP-AO has a much larger stroke. So what I didn't show here today, so we also tried some aberration correction through the thin skull, and actually there the Boston mirror goes out of dynamic range. While you still you have much larger stroke in the ALP-AO. What I do observe that there's stronger mode coupling in the ALP-AO than for the Boston.
Yeah.
Okay, so already some answers to questions. Are there any more?
Yeah, it's can I ask more? Sorry, I didn't raise my hand again.
Sure. No, go ahead.
So I think you mentioned, maybe I misheard. I didn't see a lot on what the actual brightness increase that you see for point objects is with correction. Can you, I think I misheard because I heard 50-fold improvement early on in your talk, but yeah, I think I misheard.
Yeah, that would be amazing. No, the 50-fold was for including the compressor. So if you wouldn't shorten your pulses again. No, it depends, but 6-fold intensity increase was the maximum currently, which we observed.
And I guess that's different.
When you window preparation, right? That's larger in the sin skull was larger, but at least for the cranial window preparations.
Yeah, got it. Thanks.
I had a question about your calibration of these deformable mirrors. It sounds hard. I'm not really a microscopist. Is there a chance for me to do that, or do you think that that is still like a specialist would be necessary to, like, would a commercial system be able to do those alignments and coupling mode? You know, it seems like these things should have been calibrated in somewhere, and you've done them, right, instead of the company.
I would say, let's say the calibration. I mean, luckily, some of the adaptive optics groups, for example, like Martin Boo's lab, and actually I can recommend you also like a nice webpage from them. It's like aormicroscopy.org. They explain a lot of the different aberration correction schemes, and they provide open source packages by now where they show you all of the components of the interferometer which you have to get, and provide also the script for the calibration. And that's, let's say, with that software, it's relatively straightforward. Of course, if you write your own calibration procedure, you have to be a bit more aware of how the device functions. That's pure calibration. I mean, then with respect to get the whole aberration correction work, I think it still needs a lot of user expertise tweaking. I think we are not there yet that you can just put a deformable mirror into every microscope and it runs. But hopefully we'll get there.
Thank you.
Did you ever implement Ben Judkevitz's, I can't remember what it's called, starts with an F. There's sort of cross-scanning of beams.
Is it F-sharp? Yeah, I think.
F-sharp, yeah.
No, I haven't tried. I mean, this is also, I mean, that's kind of what we wanted to have it really robustly working, right? And that's why we also, in the end, chose a relatively easy and straightforward optimization algorithm. Just assume your mode to be independent, but this is really robust and really sample independent, right? So we can really do the aberration correction of functional astrocytes, which would, for example, be really difficult with F-sharp technologies, which still require a stable signal. But I haven't tried.
Hey, Lina.
In what sense?
Sorry.
Okay, I mean, maybe Kasper, if you have a follow-up question, we do that first, and then.
I don't understand in what sense F-sharp requires a more stable signal than the orthogonal modes measurement.
It's the number of measurements basically over which you require your signal to be stable. I'm not entirely, I would need to look it up again in the paper. So let's say how many measurements or iterations you would there need to be acquired, but you have a longer measurement time, let's say.
Yeah, I think an iteration for them.
3 measurements, right? That's the kind of advantage there, yeah.
Yeah. I think an iteration for them is a 3D image that they then have to do basically some kind of. I don't think it's phase retrieval, but it's kind of like phase retrieval in my dim memory of that paper.
Yeah, I mean measuring exactly the electric field. So like they have phase stepper, yeah.
So Lina, I have several questions. So sorry, I just introduced some noise because I switched from my cell phone to my laptop. So my first question is, when you do the motion correction, so you use FPGA to measure the motion, and how can you compensate the motion in real time?
No, it's not. The FPGA doesn't. So the FPGA just processes the ECG signal, so the electrocardiogram from the mouse, and that's analyzed in real time. So it's a simple thresholding algorithm which detects the R wave peak. And then it always analyzes the 10 last heartbeats to predict when the next heartbeat will be, to generate a high-low voltage gate. And this we use to gate our microscope. So we're using scan image. So basically the gate modulates the laser clock so that the laser clock is only passed when the gate is high. And so in between, all the duct task is stopped. So the scanners stop where they were, image acquisition stops, and so on. So the FPGA is for the real-time analysis of the electrocardiogram.
I see. So then when you do the motion correction, what's the frequency of the correction?
So the heartbeat is generally, I think, 10 hertz. And then it depends on your acquisition settings, right? So if you do fast scanning, you can maybe acquire a full image. If you have a lot of pixel averaging, so if you acquire very slow image, low frame rates, you would have your image divided into several heartbeat cycles where you stop and continue. I don't know if that answers your question.
Yeah.
So that really depends on the acquisition parameters, but that's also the nice approach that you're independent of the acquisition parameters. You don't require, for example, there have been some approaches where you always trigger the beginning of the frame, but that requires that your frame is acquired within the heartbeat, while we can basically stop our image acquisition wherever we want.
Yes, thank you. So my second question is, you use Alpo deformable mirror and the Boston deformable mirror.
Yeah.
So did you see the difference in performance, or like pretty much the same?
I mean, that's similar to what I've mentioned before. I would say they are pretty similar. There is a larger, I would say, non-linearity in the Alpao mirror than in the Boston. And from the stroke, which you can have, they are different, right? So if you want to be very flexible with that respect, or if you, I mean, also people are using it, for example, like fast remote scanning, so to apply large defocus, probably then the power mirror, as it has larger stroke, would be more ideal.
I see.
But they are both continuous membrane deformable mirror, so we didn't take another, let's say, segmented mirror or so.
Mm-hmm. Did you see in your experiment the amount of aberration increased with the penetration depth? So when you image deeper, did you see more aberrations?
Yeah, we generally have that trend.
So can you give a rough idea? So like when you image at 1 millimeter compared to image 500 micrometer, so how different the aberration is?
I haven't analyzed, I would say intuitively twofold increase in amplitude, but I would need to look more into this to give a real value there.
I see.
Let's say peak to valley, right? So I mean, if we go back, I think in the hippocampus image, the peak to valley is 6 microns, but that's also really on the edges, while the center, the aberrations, they I mean, at least I find they just vary a lot from mouse to mouse, so from sample preparation to sample preparation. But we generally have a 2 micrometers peak to valley in the cortex. Let's say it's at layer 6.
Cool. So my last question is: so when you image at 1 millimeter or beyond, so what's the effective area? So when you get one correction pattern, you apply your pattern, right? For the imaging. So what's the effective area of your correction pattern?
Yeah, so the isoplantic patch definitely decreases with imaging depth. So I would assume it beyond a millimeter depth. So it's around 30, 40 microns. Again, depends on the sample preparation.
I see. Thank you very much.
Mm-hmm.
Okay, now lots of questions already. Are there any last ones? We are almost at the hour. If not, then thanks a lot, Lina, again, for a fantastic talk, and thanks for everyone for this engaging discussions. And I hand over back to Ulrike, maybe for some last announcements, outlook for the next seminars, I guess.
Yeah, so we've actually planned out our next sessions from March until August. So I'm actually very happy about this. All the information can be found on the Optical Interest Group website on our janelia.org. And the next session is actually will be given by Alison Tebu. She's a new group leader here at Janelia, and it's also about tool building, but slightly different tool building. This time it's not about the hardware part, but it will be again about dies, so the essential part more or less, which are needed in order to get the scopes running. And I think I speak on behalf of everyone to say that we're actually very much looking forward to all these 6 upcoming sessions. But again, I also want to thank a lot Lina for her presentation as well, because I know that it must have been very stressful after like submitting a paper, finishing the PhD, and right now also already giving her giving a professional talk in front of a larger international audience. So thank you very much for agreeing. And yeah, and I'm very much looking forward to meet all of you on March 2nd, as I've mentioned, for our next round and for the first talk of the next round. Okay. Then bye-bye. Bye.
Thank you. Bye.
Bye, everyone.